All right, welcome back. This is Protein Chapter Lecture 2. In our last lecture, we were talking about how DNA makes different types of proteins in our body and that different cells express these different proteins. So, for example, our skin cells will express different proteins in comparison to the cells in our blood or in our pancreas. For example, the pancreas cells will express the insulin gene, which will make the product insulin. And this is in response to our physiological needs. For example, if we get a cut in our skin, this causes bleeding, and this causes the production of various types of proteins that clot the blood. And you only need these clotting factors when you get a cut. So you want to be able to regulate how much protein you need it, when you need it, in response to both the environment and physiological need. Also, if you consume a lot of dietary iron, for example, the gene for ferritin is a protein that stores iron, is also expressed, so we can store excess iron in this way. Now keep in mind, each cell in our nucleus, each cell in our body contains a nucleus, and each of these have DNA, which has all the instructions for all the types of different proteins that our body wants to make. But not all of it is expressed. Only certain cell types will express certain types of genes depending on the need. Then the next topic we need to talk about is protein turnover. Our bodies are constantly requiring new proteins to function properly. And so protein turnover involves two processes: the synthesis of new proteins and the degradation of old ones. And the reason we do have protein turnover in our cells is that we need to be responsive to changing physiological demands of different functions of our body. So we need to make protein when we need to make it in response to, say, hot or cold or to a cut, and we need to be able to degrade the protein as well. So, for example, in our skin cells that only lives for 30 days, we must continually be replaced, and the amino acids and the proteins in our skin cells need to be continually made and degraded as a result of this. So protein turnover, as I said, is involved in two phases, the synthesis and the breakdown. So where do we get amino acids? So in our bodies, we have an amino acid pool, and that comes from two sources. One is the foods we eat, and the other is the breakdown of the cells, and so that those amino acids are recycled back into the amino acid pool. And this amino acid pool is used for five things: the synthesis of non-protein compounds such as creatinine and serotonin. Again, the synthesis of new proteins such as enzymes, antibodies, and various components of cells. The third function of this amino acid pool is the synthesis of fat from amino acid carbon skeletons. Such can be stored in adipose tissue. The fourth thing is the synthesis of glucose from amino acid carbon skeletons that can be used for energy. And last is the energy produced from amino acid carbon skeleton. And any excess of the ammonia can be excreted out via our urine via our kidneys. So what is the organization of proteins, and how does protein function become determined? Well, proteins have four levels of structure. The primary structure is the sequential order of amino acids. And the secondary structure is the spiral shape that's due to the hydrogen bonding and the sulfur bonding. And then you have your tertiary structure and quaternary structure, which we'll look at in a moment. The tertiary structure, you can think of it as the unique three-dimensional shape of the protein, a globular or fibrous structure. And the quaternary structure is the assembly of these subunits that are from the tertiary structure into another higher level of structure. So from this slide, you can see that the primary structure is mainly a string of the amino acids put together. Then this string of amino acids is coiled around in a secondary structure, which then these coils are held together by hydrogen or sulfur bonds. And then you have a tertiary structure, which is basically the 3D structure of all these coiling and intertwining of ribbon of these amino acids. And this forms like a big kind of like ball, globular ball, or subunits that can be then assembled into a higher level structure, which is a quaternary structure, as seen in this actin microfilament diagram. So protein function is determined by its shape and how the amino acids are assembled. And proteins can lose their shape by a process called denaturation. Denaturation is when proteins have lost their shape and lost their ability to function. And how do proteins do that? Well, if you heat something very highly, for example, if you think about an egg, which is basically protein white, albumin, and you throw it on a skillet, it goes from clear to white, and you're basically denaturing the protein at that point. These also acids and bases will denature proteins, heavy metals, and alcohol. Denaturation results in the irreversible loss of the protein function, and the basic takeaway for proteins is that the protein shape determines its function. So without your shape being the way it is, proteins don't function the way it should. So for example, if you look at your heme protein from your blood cells, that gives you the color red, it's made out of 4 subunits, okay, one here, and the heme group is stuck in here. So you have 2 beta and 2 alpha chains, and them assembled together gives you one heme molecule. Okay, there are 2 types of proteins in your diet. There's incomplete proteins, and these are proteins that do not contain any essential amino acids in sufficient quantities, and this can lead to growth and health being compromised, and these are considered low quality proteins. Then you have complete proteins, which are basically efficient amounts in all 9 essential amino acids, and these are considered high quality proteins.